Welcome to the Learning Pines YouTube. We hope this video helps.
You start by confirming prerequisites. At least 10 mature plants and extra seeds ready, because you will sacrifice or cross them. First, pick 2 parents showing the chocolate gene or dark color markers. This increases odds. Once done, prepare a soil plot with a 0. 5 fertilizer boost and steady environment, no temperature swings. You then perform controlled cross-pollination or seed grafting immediately, labeling each attempt. Keep detailed notes so you can track which crosses produced chocolate offspring. Next, you execute the complex middle steps. Perform targeted crosses, chocolate-bearing plant with a dark variant, repeating 5 to 10 times to stack probability. Then move seeds into incubation at 22-24 degrees with roughly 60% humidity. Stable conditions reduce mutation failures. Make sure to label trays and rotate light cycles to mimic natural pollination cues. Monitor seedlings every day, removing weak plants and keeping the healthiest candidates for a second-generation cross. Finally, you choose the top 2 plants that show chocolate expression and perform the final cross to stabilize the mutation, then let seeds reach full maturity before harvest. Once mature, store seeds at consistent 4-6 degrees to preserve viability. Make sure to perform 1 more generation to confirm stability. If you don't get the mutation, don't despair. Increase attempts by 2 times, recheck parent genetics for the chocolate marker, and double-check environmental steadiness. Temperature and humidity are common hidden culprits.
Thanks for watching. Consider subscribing if we helped you.